Now, here are the scientific facts. Arthur David Skerland explains the unimaginably intricate and specific steps that would be required to produce a living system. Now, this is from the scientific viewpoint. If you were to create life in a test tube, this is the scientific procedure that would be necessary. The origin of life by natural processes would require the following steps. Number one, formulation of simple building blocks such as amino acids and nucleotides, because they're a part, that chemistry is a part of the DNA. So you got to have these simple, basic building blocks. Number two, arrangement of these molecules into biologically important compounds such as proteins and DNA. But remember from the introduction, there's a hitch. If you have the proteins, for them to function in a cell, you got to have the DNA too. And if you have the DNA, you got to have the proteins. So you have to arrange these molecules into biologically important compounds such as proteins and DNA. It's pretty hard to do. Scientists haven't been able to do it. Number three, assembly of these proteins into a metabolically active system and organization of a complete, independent, stable, self-replicating cell. Wow! So you start with the building blocks, you form them into proteins, and DNA is a protein. Then you assemble those into a self-replicating cell. All right? Number four, introduction of additional genetic information to form new and complementary life forms. Not just enough to have frogs in ponds, you got to have little boys fishing for those frogs. It goes together. Seems like I remember that. Skerland then details why this is beyond the realm of the possible. Most of the cell's important functions are carried out by compounds called proteins, which are a chain of amino acids, 20 different types, linked together. The protein's characteristics and functions are determined by the number and particular arrangement of specific amino acids. Every protein in a living cell, so important. Every protein in a living cell uniquely consists of entirely left-handed amino acids. Amino acids come in two forms. Left-handed, which fits right up against right-handed. Amino acids come in 50% each way. But all living proteins are made up of only left-handed amino acids. Oh, that's important. The products from the primordial soup experiments have been a mixture of a few right-handed and left-handed amino acids. Okay, we got amino acids. We're about to create life. Wait a minute. Got a few right-handed, a few left-handed, resulting in approximately 50% of each. If only one amino acid is changed, malfunction, right, the results, the soup is spoiled. Just one, and it won't work. Wow, God hemmed life in. Textbooks teach that Stanley Miller performed an experiment that produced the chemicals of life. However, that's not the case at all. Authors Gish, Parker, and Bliss explain that one of many insurmountable problems surrounding the experiment, and a vital part of Miller's apparatus, is a cold trap. Important. He put a cold trap right there at the bottom, so that when he got the amino acids he wanted, he stopped them, took them out. So he said, I got the amino acids. Well, now wait a minute. If you keep running the experiment, the energy that created the amino acid also destroys them. Ignore that. Destroyed by the same energy, but that's not all. In that cold trap, it's important to realize, even with the cold trap, the amino acids were equally left-handed and right-handed, resulting in destruction of any protein that might conceivably arise. The soup is spoiled again.